Okay, after watching this video, you should have some understanding of how splicing works. The chemistry has already been reviewed, those two transesterification reactions, but the other real mystery of intron removal is how does the spliceosome find these precise locations? How is the branch point A identified, and the 5' site and the 3' site, and exactly how is the catalytic apparatus assembled? These special sites, the 5' site, the branch point A, and the 3' site, they have a characteristic sequence features that are recognized by small nuclear RNAs. Each small nuclear RNA is listed here in table 30.3, and the role that they play in splicing or intron removal is listed in the table. There's always an RNA molecule that's the central part of the SnRNP, and we call it particles because there's also proteins associated with these RNAs. So the U1 SnRNP has a U1 RNA, 165 nucleotides in length, and its role is to bind the 5' splice site. I don't know why they say 59. This should read 5' splice site. U5 also binds the 5' splice site and the 3' splice site, and then U2 and its associated RNA binds the branch point site. One of these recognition events is shown here on page 1008. You can see here base pairing between the U1 SnRNA and the 5' the nucleotides at the 5' splice site. These are complementary to each other, such that we get recognition of the 5' splice site by its base pair complementarity to the U1 SnRNA. The U4 and U6 SnRNPs have to do with the actual catalytic process, and the whole process is outlined here in the figure 30.38. We see here the sequence of events. First, U1 and U2 SnRNPs find the branch point A and the 5' splice site. And now we bring in the U4, U5, U6 complex. So all three SnRNPs are assembled as they join this complicated structure. And the catalytic unit is the U6, the U6 SnRNP, but it is its catalytic activity is masked because it is in complex or base pairing with U4. So what has to happen is that U1 and U4 have to dissociate from the spliceosome during this next step, and that allows U6 and U2 and to interact productively with each other to initiate the first transesterification reaction. And then U6 and directs now the transesterification of the second reaction, so that the exon, the 3' hydroxyl of the first exon, can now attack and make the spliced exons. And then the lariat intron is carried off to be degraded and then the whole process this has to disassemble so that the units can be recycled for the next intron removal sequence. So in biochemistry, we talk about enzymes that are obeying different models for substrate recognition and activity. One is the so-called lock and key model, where the enzymes have a pre-existing rigid active site, and the substrate just fits perfectly into that active site like a key into a lock. And then there are induced fit model for enzyme substrate interactions, where substrate binding changes the structure of the enzyme a little bit. We get some molecular motion associated with substrate recognition and catalysis. And that those type of enzymes are the ones that we have previously been studying, like myosin and ATP synthase and DNA polymerase. But the spliceosome is a completely different type of model where the enzyme is being sort of built into, and the substrate, the intron, becomes part of the catalytic apparatus, providing the chemical groups needed for catalysis. And there's a lot of remolding and restructuring of the enzyme, especially at this critical step where U1 and U4 leave the spliceosome. And this process is driven by ATP hydrolysis. And it using helicases to separate the U1 from the 5' site and separate U4 from U6.